Hello, and we are going to talk about patterns of inheritance today. This is chapter number 9, basically Mendel's materials. Genetics is a study of heredity, and as genetics explains why offspring of purebred dogs are like their parents. Inbreeding of dogs makes them some genetic disorders more common, also makes some of the things that we like about those dogs a little bit more common. And again, a dog's behavior is determined by both its genetics and its environments. And there's a prerequisite number of cute dogs. So basically what we're going to talk about today is heredity, which is just the transmission of traits from one generation to the next. One of the pioneering founding fathers of this is Mendel, and there's his picture. Nice looking guy. And he performed a lot of different experiments on a really kind of interesting and cheap model organism, which is basically the pea. And what he was looking at were traits like flower color, pod shape, pod color, stem length, etc. So he looked at a number of these traits that everyone would possibly agree on, and so here's a flower color, that's obviously white. And what he did are a bunch of what we call monohybrid crosses. That's where he crossed two of the same particular things together to see what happens from them. And sorry about that. And then, so again, if you cross a dark flower with a light flower, he actually got all dark flowers. And then when he took this generation and crossed them together, he got a ratio of 3 dark flowers to 1 kind of white flower. And so what Mendel did here that was very interestingly is that he came up with this idea of alleles. And actually, let's go. He came up with this idea about alleles. Came up with the idea about alleles, and he came up with this idea about the alleles, and that an allele is an alternate form of gene. So we have a gene, what we saw before is we have a gene for color of the flower, but one allele is dark and one allele is light. And what he said is that an organism is homozygous, the same if both alleles are identical, and an organism is heterozygous if those alleles are slightly different. And so when we have these different alleles, one allele can be dominant, one allele can be recessive. The dominant one is what always appears, and the recessive one only appears when it is homozygous. What he was saying that gametes only carry one allele from each inherited characteristic. So the sperm or the egg, or in the plants of a flower, the pollen or the egg can only carry one trait. And we talked about that before, is that if you only get one chromosome, one of each type of chromosome from your parents. And what he said that this is basically the law of segregation, that two members in an allele pair separate from each other during the production of gametes. Okay, and he used something that we call the Punnett square. An organism's phenotype is what its physical traits are. Its genetic traits are what we call its genotype. Homologous chromosomes have genes at specific loci, and alleles are the same at each locus. So what the idea is here is the homologous chromosomes, here's the maternal chromosome, here's the paternal chromosome, and that these are genetic loci, which are physical locations along this chromosome. So if we look at the shape of this chromosome that we have here, and again, we can have a dominant allele and we can have a recessive allele. The dominant allele is indicated by this B, and a recessive allele is indicated by this lowercase b here. And what we call a dihybrid cross is when we cross something with two traits. And so in the peas, if we cross a yellow smooth with a green round, we would get all yellow smooth. So who is dominant here? And then when we cross these together, these two heterozygotes together, we get a number of outcomes here. And we'll have a lab in about a week and a half to actually look at this. And so this is actually real and occurs within the peas. And so in Mendel's dihybrid cross support the hypothesis that each pair of alleles segregate independently from others during, this is what we call the law of independent assortment. And so on the allele that we looked at the other time, the A, B, and C, they would each assort independently from one another. And so even though they're on the same chromosomes, they can do this. Laws of probability is that, again, Mendel had a strong math background, is that the rule of multiplication states that the probability of a compound event is a product of separate probabilities of each independent events. And so you have a 1/2 chance of getting something and another 1/2 chance of getting something. The total chances of you getting something are 1 in 4. There's the multiplications of those. And again, we just kind of talked about this. Again, a pedigree is just our way to kind of look at this. And so here's looking at a number of people, and we have a number of different phenotypes that we have in there. And then dominant traits are not necessarily what we call normal or more common in there, and that's basically what we look at that. And so this dominant recessive doesn't necessarily mean good or bad. Wild type traits are those that we most often see in nature. And again, they're not necessarily the most dominant alleles. A family pedigree just shows the history. And so in this pedigree, we have boxes representing males. We have circles representing females. If we put those boxes out, and then we have these filled-in boxes give you what the phenotype is. And so in looking at this, we can see that that attached ear is probably recessive because it skips this generation right here. And so that's the main thing that we're looking for. Beautiful looking family, the attached and detached ears. Human traits, basically many human traits, so really simple inheritance patterns. And so they're controlled by a single gene on an autosome. And here are some of the different traits that we look at: albinism, cystic fibrosis, phenylketonuria, and there's a number of these different traits. Most human genetic disorders are basically recessive, and then individuals that have a recessive allele appeal normal blood are carriers of the disorder. And so you have parents and offspring who can be totally normal. 1 in 4 of their children, or in this case here, are going to be deaf because they carry that recessive order, and the double recessive individual will be deaf. Deaf, not deaf. Cystic fibrosis. We can also see that as well. Huntington's disease and and so again, there's a number. We look at this dwarfing. So we see DD, small d, and actually this is kind of one of those things where if you see double D is actually a lethal trait, and this does not occur. And so again, because this is a dominant trait, you can see that. Genetic testing is what we see a little bit. Now, Mendel didn't have it all right. I mean, he figured a lot of stuff out, but then there's always exceptions. And so we have something that we call incomplete dominance, F1 hybrids. And so if we cross something with a red flower and a pink flower, red flower and a white flower, we get a pink flower. And in this case here, we can see what the heterozygotes are, and it's kind of fairly interesting in there. Blood groups are the same way that we can see, is that A, B, and O. A and B are codominant with one another. O is recessive. And so, again, what we see with the A phenotype, again, is because it's dominant. The AB phenotype, we have both of those. And basically, you're expressing a protein that is figured, and you can always see that there. And so we'll talk a little bit more about this biochemistry. And so the reactions when blood groups left, and so if you have antibodies are present in blood, and so you're going to have anti-B antibodies, and so you won't react with O, you won't react with A, but you'll react with B or AB. If you have anti-A antibodies, you react with A, you react with AB, but you won't react with these others. If you have no antibodies, you won't react with any of these. If you have anti-A and anti-B, you will react with everything but O or 0. Okay. Skin color gets more complex because you're looking at 3 different genes, and basically what your skin pigmentation is due to is basically combination of all these genes, and so it's really hard to take a look at that. Polygenic inheritance is additive effects of two or more gene types. And then we look at here identical twins, yet they can still look a little bit different. All of us know about the heavy twin and the fat twin, and so our basically our environment can also affect how we look as well. Many human being characteristics are a combination of hereditary and environment. Only genetic influences are inherited. So again, Arnold Schwarzenegger's kids are not going to become muscular due to his musculature. Okay, and so this is basically going through these materials, and we will have a lab on this in a little bit. The chromosomal basis of inheritance, basically it's chromosomes, what we inherited. It's chromosomes that undergo segregation, independent assortment through the laws of membranes. We will try and do this. Here we have some insects, and then if you hop into insects, linked genes are genes that are really close together on the chromosomes in Drosophila melanogaster. There's a sex linked that we have in there, and so if these two genes are located so close together that we never really have a crossover event between those, and so relatively rarely. So again, this is one of the things that we need to know is what is a crossover event. Crossing over can separate linked alleles, produce gametes with recombinant chromosomes, produce offspring with recombinant phenotypes, and so hopefully in the next thing we'll get it. Recombination frequency is basically an idea of how far apart these are due to the number of recombinations that one of us. And so Thomas Hunt Morgan's did that. What we want to do is look at, so here we have 22 chromosomals and one sex-linked trait. Females can only give their X-linked, and so basically that's one of the reasons why we have 50-50 chance of having females or males is due to the presence of the male. And so she is not at fault when she has either all males or all females. Here's an example of the chromosomes, Y chromosomes, the Y chromosome here and X chromosome here. You can see the difference in their size. There are some genes that are located on the Y chromosome or on the X chromosome that we tend to see a little bit more. One of the ones that we really see is color blindness. Sorry, that's in my 12 minutes that are up. And so here's one of the tests that we have for color blindness. Hemophilia is an example of that, the Tsar of Russia. And then again, we look at evolution. Here's looking at the dog.